Okay, good afternoon, everyone. First of all, I would like to thank the committee for selecting me to give this flash talk. Alzheimer's disease is a progressive neurodegenerative disorder characterized by gradual loss of cognitive abilities with cortical and hippocampal atrophy, with synapse and neuronal loss, and neuroinflammation. Pathologically, there are two major hallmarks: the extracellular accumulation of amyloid beta plaques and also the intracellular accumulation of neurofibrillary tangles formed by the aberrant hyperphosphorylated tau protein. The interesting thing is that the cognitive decline correlates well with the increase in tau levels and also with the synapse and neuronal loss. Alzheimer's disease can be classified into two groups according to the onset age. First, the late onset, also known as sporadic Alzheimer's disease, and second, the early onset, also known as familiar Alzheimer's disease, that is caused by dominant inherited mutations on APP, presenilin-1, and presenilin-2 genes. Interestingly, mutations on presenilin-1 genes are also related to frontotemporal dementia that are also characterized by tau pathology. Presenilin are part of the gamma secretase complex, which is the enzyme implicated in the processing of APP by the amyloidogenic pathway, and they're responsible of the amyloid beta generation. Therefore, mutation on these genes cause an alteration in the processing of APP, increasing the levels of amyloid beta. However, the mechanistic link between presenilin and tau is still unknown. As we consider this mutation as a loss of function, our hypothesis is that presenilin deficiency contributes to tau hyperphosphorylation, aggregation, and overspreading in Alzheimer's disease. To do this, we have used the presenilin conditional double knockout mice described by Dr. Calasaura, in which the presenilin-1 gene is deleted only in glutamatergic neurons of the adult brain because of the expression under the kinase 2 alpha promoter. We have seen that these mice at 9 months of age present behavioral abnormalities like an irregular body posture and the inability to extend properly its limb in the hindtail test. And also an age-dependent neurodegeneration measuring the wave brain that affects both the cortex and the hippocampus. And this neurodegeneration is not present from the birth because there are no significant differences at 2 months of age. Next, we analyze the tau pathology using 3 different well-characterized antibodies against different phosphorylated residues of tau protein, the CP13, the 88, and PHF1. We saw that at 9 months of age, only total presenilin deficiency caused an increase in phosphorylated tau levels, both in cortical and hippocampal lysate, without altering the total tau levels. We confirmed this result by immunohistochemistry using the CP13 antibody. We saw significant increase in phosphorylated tau positive cells in the presenilin conditional double knockout mice, especially in memory-related areas such as the entorhinal cortex or the hippocampus. Colocalization analysis between phosphorylated tau antibody, the CP13, and different cell markers revealed that phosphorylated tau were not only accumulating in neurons, and it were also present in microglial cells, oligodendrocytes, and astrocytes. This result was also accompanied by an increase in GFAP and EVA1 levels, suggesting that presenilin deficiency caused neuroinflammation. Then we wanted to check the effect of presenilin deficiency on human tau protein, as it seems to occur in familial Alzheimer's disease and frontotemporal dementia-linked mutation. So we crossed our presenilin conditional double knockout mice with a tau transgenic mice that overexpress the human tau protein with a mutation linked to frontotemporal dementia. We performed a similar biochemical analysis than before, but at 6 months of age, and the first thing that we saw was an obvious increase in the tau transgenic mice compared to the non-transgenic, and also an increase in phosphorylated tau levels in the presenilin-1 and especially in the double knockout mice with tau expression compared to the tau transgenic mice. Then we use the PGFI antibody that is against a specific phosphorylated residue of tau that is only present during the development and in pathological condition. And we saw a specific band that were only present in these 2 groups, but not in this control, the tau transgenic mice. Then, using the MC1 antibody that is against a pathological conformation of tau, we saw a similar result than before, an increase in these 2 groups, but not in the tau transgenic mice, suggesting an increase in phosphorylated and aggregated tau. We confirmed this result by immunohistochemistry using the PGF5 antibody. We saw a specific staining in the presenilin 1 and especially in the double knockout mice with tau expression, but not staining in the wild type or the tau transgenic mice. Again, in memory-related areas, such as some areas of the cortex, the amygdala, or the hippocampus. And a similar result with the MC1 antibody, we found an increased levels of aggregated tau in these two groups, but not in its controls. Then to study the aggregation pattern of tau in deep, we use synchrotron infrared microspectroscopy analysis, and we found increased pathological beta sheet structures, concretely in the beta intermolecular and beta antiparallel protein structure, in the presenilin conditional double knockout mice with tau expression compared to the wild type, and especially in the corpus callosum of these mice. Histological analysis of the consecutive slide used for the synchrotron revealed the presence of MC1 positive cells and also the presence of Congo red positive cells, suggesting that this pathological beta sheet structure might be related to aggregated tau. Then, to check the effect of presenilin deficiency on synaptic pathology, we perform a synaptosomal purification to isolate the synaptosomal, the pre- and the postsynaptic terminals. We saw that presenilin deficiency increases the phosphorylated tau level in both in total lyset and also at the synaptic terminals, and in correlation with a clear decrease in synaptic markers such as PSD95, taxin-1, synaptophysin, or beta-actin. One possible functional consequence of this pathology could be an hippocampal memory and learning deficit. So we performed a Morris-Water maze and a contextual field conditioning, and we saw that presenting conditional double knockout mice with tau expression has increased hippocampal memory and learning deficits. So as a conclusion of this part of the talk, neuronal presenilin deficiency causes behavioral abnormalities and age-dependent neurodegeneration associated to cerebral phosphorylated tau and neuroinflammation. Loss of presenilin function increases phosphorylated and aggregated tau in human tau transgenic mice, and presenilin deficiency exacerbates synaptic pathology and hippocampal-dependent memory impairments in human tau transgenic mice. This result has been recently accepted in this paper, and now I would like to briefly talk about which could be the molecular and cellular mechanisms implicating this pathology. We have focused on autophagy, one of the main tau elimination pathway, and we have seen that presenilin conditional double knockout present an increase in the LP3 ratio, especially at 6 months of age, and correlating with tau pathology. This result was also accompanied by an increase in P62 levels, suggesting an increase and accumulation of autophagosome in these mice. Then we checked also the levels of some proteins involved in the autophagy pathway, and we found that presenilin deficiency alters the activity of some proteins such as mTOR or AMP kinase. Then we wanted to know if we could replicate these results in vitro, and using primary neuronal cultures, we found that presenilin deficient neurons has also an increase in phosphorylated tau levels and also in the LT3 ratio levels. And finally, to see if we can have the same result, but in using the presenilin 1 familial Alzheimer's disease linked mutation in collaboration with Dr. Carlos Vicario group, we have used the fibroblasts from patients with presenilin 1 familial Alzheimer's disease linked mutation. We have seen that the fibroblasts of this patient doesn't, when we block the autophagy with chloroquine, doesn't respond at the same level as the control fibroblasts do, and also accumulating APP. So this preliminary data suggests a link between presenilin and A-beta tau pathology through autophagy. Finally, I want to thank all the people from my lab and also the finding, and thank you for your attention.